Hey, good evening, everyone. I hope you are having a fun time with molecular evolutionary genetic analysis tool. I hope you have downloaded this current version, MEGA 12, and it's been installed. So today we will see how to use AI integrated in MEGA tool. I hope you guys have seen this MEGA-GPT that is been already integrated in the MEGA version 2. So today we will be using this MEGA-GPT to understand the variations in biological information or DNA sequences to be precise. So we will see various examples. Okay. One such example here is just generative pre-trained transformer. Even though it looks like ChatGPT, it's not at all it's a language model, but much more specific for molecular evolutionary genetic analysis. Okay, so this is as advertised. It's been built on a retrieval augmented generation framework, and they use a hybrid approach. Unlike ChatGPT, you will see they will not hallucinate, and they use a hybrid approach, which is much more specialized for static training data, and they definitely reduces domain insensitivity, especially to biological data, and also they reduces that is literally nil hallucination. So it has been released in 2023 last year. I hope this model, very less citation has been achieved so far. It's not even 12 months. So I feel anyone who integrates this model in their biological research, their paper will stand out in reviewers and editors' point of view. So getting into the topic, see what happen if I want to analyze a pathogenicity score for a particular gene. In order to just see if the gene mutates, will it become a disorder to a biological system, or they are just a neutral mutation? If I ask this question to ChatGPT, okay, does not directly provide any pathogenicity score, which is absolutely lie. Okay, molecular evolutionary genetic analysis tool have a tool to provide this. Okay, so, but you see, when I use the same question or prompts in MEGA-GTP, they give me a clear-cut pathway of how to do this analysis. Okay, so I just want to demonstrate how to predict the variations in biological data using MEGA-GTP today. So when I ask this question, say especially we will be using this CFTR gene today to understand how this MEGA-GTP function. So regarding this link mega gdp, I will give this link to your this is a link you can access this ai tinyurl.com/gdp-mega. I will be circulating this link in your group today, and you after seeing this demonstration, please use this link. Okay, so we want to analyze the variation among a protein coding gene, and this is a prompt given. Okay, mega molecular diagnosis md is a new function that has been integrated along with the machine learning tool in mega 12 version, which is not present in previous versions. So we will see how to use this. So this is a pathway how to go and access this gene. Sorry, I firmly believe that by now you guys have know how to prepare your input file, how to do a multiple sequence alignment, how to import that into Mega, and how to carry out multiple sequence alignment if necessary, and also phylogenetic analysis. And I have most of the people have constructed a high quality phylogram, and congratulations for that. And I'm very sure and I'm very confident that by now you guys can handle biological data through ClusterLex and Mega. So now today we will see how to uh, test this prompt given by MEGA-GTP in Mega Software Tool version 10, 12. All right. So before getting into this molecular diagnosis mode, this is an option we will be using. I want to. I hear that many of bachelor's and master students, they are finding it little difficult to follow the vocabularies. So we thought today we will do some vocabulary check before we get into the actual prompts. Okay. So what is this? This is an example gene I'm taking today, CFTR. Why this gene? This is cystic fibrosis transmembrane conductase regulator. They are present in most of the epithelial cells, membrane channel in the membrane of the epithelial cells in most of the animals. They are responsible for maintaining hydration and, in many of the specialized organs, including lungs, pancreas, intestines, sweat glands. If this protein fails, that's when we have cystic fibrosis. Okay, so let's see how this cystic fibrosis has evolved. This gene has evolved across various phyla, and let's test using MEGA-GTP. If we mutate a particular amino acid in this gene, what will be the effect? So effect can be of only two consequences. Is that a deleterious effect which ends up in cystic fibrosis, or it is a neutral effect that does, even though there is a mutation, it does not show any effect. So this is the only two consequences it can be. So let's see how to use this. Before understanding that, I want to refresh your memory. Codon table, three nucleotides code for one amino acid, because we are going to study about the mutation in the amino acid. So this is a first codon row, second codon, and third codon. I hope this is a very basic. You guys know about this. I want to refresh your memory onto this terminology: synonymous mutation and non-synonymous. When there is a synonymous, even though there is a substitution of A to T, there is no change in the amino acid. In non-synonymous, amino acid definitely changes. In non-synonymous, there are nonsense, missense, there are various mutations, but for now, we should be very clear about the differentiation between synonymous mutation and non-synonymous mutation. Okay. So in non-synonymous mutation, whenever we say pathogenicity impact, it means that substitution of a particular nucleotide ends up in changing the amino acid, which may usually when they are present in the conserved region, they have a deleterious effect. So what is this conserved region? Let's understand that. When I hope you guys can recognize this. This is multiple sequence alignment. Okay, any biological data analysis will start with aligning the DNA or amino acid sequence as a multiple sequence alignment format, and from there the analysis starts. Once you do the multiple sequence alignment format, you should know these four vocabularies. Okay, conserved site have same nucleotide of amino acid at a given position. So here, the examples of nucleotides given here, you should read it vertically. Okay, at a particular position, what is the sequence? That's what they, for example, this AAA or 5As, they have been represented like this. Okay, so conserved site don't have any variation, whereas variable site will have a variation at a given position in an alignment. All right, parsimonious site, at least two characters occurring more than two times. For example, that is a variation. Or in other way of saying, there are only two types: conserved site, variable site. Variable site can be reclassified again into parsimonious site and singleton site. If the variations are more than, if there is just one, it is singleton. If it is more than one, it is parsimonious. Okay, so if we want to know how this is applied practically, all primer designing usually happens in the conserved site. Okay, across the phyla, if you want to design a PCR primer, they usually comes from a conserved site. Variable site actually define the uh, particular function of the gene in the phyla. Okay, so these are the just to refresh your memory. So in order to assess the pathogenicity or changes of amino acid in the particular organism, will they impact the organism in disease and disorders? We have a score called pathogenetic assessment score. They are actually calculated by various means. We are going to take these three means. Okay, evolutionary diagnosis, polymorphism phenotyping. V2 is actually a version 2 of a machine learning that is being used by the Mega right now. Shift is a sorting intolerant from tolerant. Here, intolerant means neutral mutation. Intolerant means actually deleterious mutation. Tolerant means we have neutral mutation. Okay, so score meaning higher the evolutionary diagnosis score, there is an impact of the disease. Okay, so meaning they are more pathogenic. So in polyphen, they are denoted by binary 0 or 1. 0 means the mutation is beginning. 1 is it is damaging. It is pathogenic. So likewise, shift is also assessed in various ways. Okay, these are the little vocabularies you need to know. So we will get into the uh, the MEGA tool now. You need to know that to understand the variability of this CFTR gene, I use this prompt in the MEGA-GTP. Okay, so MEGA-GTP, I will give the link today. You can check it out. So the MEGA-GTP told me to open MEGA-MD molecular diagnosis tool where machine learning has been incorporated. Then I will be entering CFTR gene information. I will navigate to the mutation explorer window, gene search, and how I'm going to study the changes in the amino acid sequence among this gene, CFTR gene. Okay, let's use MEGA-GTP now. Okay, I hope everyone have installed the current version, that is 12. So diagnose mutation. Here, do you wish to specify a coordinated information of variants of the interest? I will go to wizard. I will open the wizard. So this mutation explorer window has been opened now. So I will press CFTR. I will go and study this gene. You see how simple it is now. This MEGA-GDP has this function actually works by connecting to internet. You need an internet connection. See, it has extracted protein ID across various phyla from the internet, from the GenBank. Okay, so this is the first result. Diagnose variant. I will click it. I'm going to click this diagnose variant, and I have this variant window. Okay, so here you are seeing C. C means conserved site, as we have seen the definition of conserved site. Here we are seeing variable site. Only when where there is a variation, you can see among this site, what are the parsimonious site, what are the singleton site. You can clearly see them. Okay, so this is not our target. We are going to study the mutation at a particular region. Okay, let me consider this particular site. Okay, among the multiple sequence alignment that is being shown here. So this is a multiple sequence alignment you have here. So I want to study this particular site because I feel they are parsimonious. So go here. Okay, here. I want to, okay, here I will go. I want to study tryptophan. At this site, if it is being substituted by tryptophan, what will be the, so let's see. It's a neutral mutation. Okay. Here you can see the polymorphism phenotype. Okay. And evolutionary diagnosis and shift value. It says neutral. If it is being substituted by 309, it will be a neutral mutation. Okay. So let's keep studying for okay, let me choose this side. It is more, looks like more parsimonious. Here again, I will go for tryptophan. Okay, let's check the results now. Okay, it is deleterious. Okay, this is definitely deleterious if it is being substituted. Okay, this ways using machine learning output, you can actually study what if a particular amino acid has been substituted in a particular site. Not only just studying that, you can make an ancestral, say for example, maximum likelihood tree. Okay, I'm going to create a tree to understand how this is, in a form of how this mutation progresses across the phyla. Okay, so before seeing the tree, we should see what are the options we had actually. Okay, you see this tree is still loading. It takes depends on your system configuration and many more factors. Okay, all right. So we have a tree. So this gene actually data are across from human, car data, and you see all the here rabbit, rat, and squig. They form a different group. Zebrafish and other fishes they are placed as out group. Okay, so wherever you see A, it means bootstrap values are agreeing. Wherever you see no bootstrap, it means that particular region it's not agreeing. Okay, this simply shows how this particular gene has been evolved due time. And if you are studying for a, in case of diagnosing a disease or something, that's how you can use this molecular diagnosis AI tool to diagnose the changes in the multiple sequence of event. Okay, so today's class is all about how to understand, initially start using Mega, which is very much specific for the assigned function. I'll be sharing this Mega's link in the group. Do visit this website and use this prompt that is being circulated in the video and try it for yourself. Tomorrow, we will be working with time tree, how to use mega function and time tree together. Thank you for joining today. Happy learning.